Hello, I'm Dr. Terry Conger. I'm with the USDA Animal and Plant Health Inspection Service, Veterinary Services. I'm the Poultry Health Specialist in Field Operations District 4. The USDA has the United States divided into 4 districts, field operations districts, and each district has a specialist in terms of poultry. So I'm covering poultry in District 4, which includes 8 states. I'm here today to discuss with you the importance of the poultry industry, not only in the United States, but the importance of poultry globally as a protein source for all the entire population of the world. So it is our goal to maintain the health and vitality and reproductivity of poultry so it can help to feed the United States. I'm going to cover 2 important diseases, the first one of which is highly pathogenic avian influenza. An overview of avian influenza viruses, that virus is in the family Orthomyxoviridae. There are 3 main serotypes, type A, type B, and type C. Type A affects multiple species, especially birds. It is infectious for all species of birds. Influenza A, or type B, affects humans, particularly seasonal influenza. And type C affects both humans and swine. So if we give a look at the different serotypes of influenza A viruses, it's divided up into 16 hemagglutinins and 9 neuraminidase antigens. They are surface antigens by which the avian influenza viruses are identified. There are 9 different neuraminidase antigens, and 16 different hemagglutinin antigens. And they arrange themselves in a variety of groups. If we look at all the possible different combinations, we have 144 different possible serotypes of influenza A viruses. Formerly, influenza A viruses were divided into 2 major groups. The first, highly pathogenic avian influenza, or the second, low pathogenic avian influenza, or HPAI and low P, LPAI. Now they're divided into 3 different groups. HPAI is primarily hemagglutinins types 5 and 7, but they can be any strain of influenza viruses depending on the degree of mortality that results from an infection in a flock. The second group is the low path AI groups, LPAI, which now are only high H5 and H7. These 2 serotypes can mutate, reassert to become high path AI, but as a low path AI, they're causing only a very negligible rate of mortality. And the third major group is all other influenza viruses, which are all low pathogenicity. Only the first 2 groups are reportable to the OIE. If we look at the different influenza A viruses, it's made up of 11 different proteins that are coded by single-stranded RNA viruses or genes, hemagglutinins, neuraminases, and the internal proteins are nuclear protein and matrix proteins. The general non-specific tests for all the Generally, the matrix proteins for all avian influenza viruses have a similar makeup. So the non-specific tests such as PCR will pick up the matrix protein, and a matrix, if anything positive for the general PCR test, are further tested at the National Veterinary Services Laboratory to differentiate whether it's truly highly pathogenic avian influenza. So, epidemiologically, the potential sources of an initial infection in an area, geographical area, one, naturally infected domestic poultry, but the primordial reservoir is migratory waterfowl or other wild birds, but primarily migratory waterfowl is the reservoir that carries the avian influenza viruses. Also, domestic pigs can be involved as a source, and companion or pet birds can also be a source for the virus in any particular area. But the primary reservoir that we're concerned with are migratory waterfowl. If we look at the transmission routes of avian influenza viruses, we see at the top ducks and geese, shorebirds carrying the viruses. They can transmit that virus to either small mammals or other avian species or birds, or they can transmit it to poultry, turkeys or chickens primarily. Once turkeys and chickens become infected, the virus can be transmitted by the movement of contaminated equipments, vehicles, service personnel, one place to another. So once it gets into a commercial poultry flock and humans pick up, can serve as the transmission mechanism from one flock to the other or another area to another by contaminated vehicles or equipment from one place to another. If we look at the wide spectrum of all influenza A viruses, like all avian species are susceptible to all influenza A viruses, but a total of 6 hemagglutinin types can be infectious for humans, as well as 2 neuraminidase types, and several are also infectious for hogs, and a few are infectious for horses. So you can see there's a wide spectrum of susceptible species when it comes to influenza A viruses. If we look at the clinical signs and pathological signs associated in poultry with a high pathogenic avian influenza infected flock, we see primarily upper respiratory signs, which would be swollen sizes and nasal oral discharges, difficult breathing and whatnot. But highly pathogenic avian influenza is a systemic infection, so it can cause neurological signs, incoordination, and it causes pathological lesions throughout the body in terms of petechial and ecchymotic hemorrhages. So once again, the general signs of avian influenza virus, highly pathogenic avian influenza are difficult breathing, oral and nasal discharges, cyanosis of head and comb and wattles, ataxia, ruffled feathers, but the primary sign is high mortality here. In an infected flock, mortality can approach 100% depending on the particular species and the particular serotype of high path AI. When in the 2014 and 15 outbreak, I performed the epidemiological survey of the 2 commercial turkey flocks that were affected by serotype H5N2, highly pathogenic avian influenza. 3 days after the first spike in mortality, when I surveyed the flock, visited the flocks, each flock displayed up to 95% mortality. These were 45 to 50 pound tom turkeys at that age range, and turkeys are very susceptible. So we had a very severe mortality when it came to these turkeys. In chickens, it's not nearly as pathogenic. Waterfowl, like I said before, are the natural reservoir host for all influenza serotypes, but not usually high path AI. Usually, migratory waterfowl will pick up the high path AI through exposure to infected chickens and turkeys, that in which it has mutated to a high path AI strain, and once it gets into migratory waterfowl, in their flight patterns, they can transmit that virus from one place to another and cause a severe outbreak. And we'll go into the 2014-15 outbreak in the United States to demonstrate that. The geographical origin of different strains may often be gleaned from their genome, but highly pathogenic Avian Influenza A viruses, H5 and H7 strains of low pathogenicity, are the only two types that are reportable to the OIE at this point. How does avian influenza get into a flock or an area? Well, the primary culprit is this duck or goose or shorebird carrying that virus here. Everywhere this high path AI causes very negligible clinical signs in migratory waterfowl, but they transmit that virus everywhere they go through the digestive tract. So that is how it's transmitted from one place to another. So the primary way in which avian influenza gets into an area or a flock is direct or indirect contact with exposed migratory waterfowl. When I say indirect contact, I mean tracking in contaminated fecal material from a pond or area, or duck hunters or goose hunters, when they harvest their ducks and geese, they their clothing and equipment can be contaminated with that virus, and they can, as a fomite, those viruses can be transmitted to poultry they come into contact with. Less important ways in which AI can be enter an area is imported or smuggled, exposed or infected birds, or, like I said before, movement of contaminated equipment or people onto a premise from one place to another. Or the fourth way is mutation of high path AI into low path AI into high path AI. So these are the four primary ways, but the primary culprit here is this duck or goose carrying that virus. If we look at the world migratory flight patterns, we have over here the eastern hemisphere and the western hemisphere. In the United States, there are four primary flyways that in the springtime, ducks and geese will migrate to south, and in the fall, when it gets colder, or opposite, in the springtime, they migrate north to their nesting grounds when it gets warm up here in the north, where they have their nesting grounds up here in Canada and Alaska. And in the fall, they migrate south because it's getting cold, so they want to go down to the warmer regions. So we have the Pacific Flyway, which parallels the Pacific coast here, or the Central Flyway, which is right over the central part of the United States, right over Arkansas and Texas, where I'm located, or the Central Flyway, which kind of follows the route of the Mississippi River, and the Atlantic Flyway, which flies down parallel to the Atlantic coast. But the key element here is there's interaction and interchange of that virus up here between the Eastern Hemisphere and the Western Hemisphere through the Bering Strait, where the migratory waterfowl mix and intermingle up here in Alaska across the Bering Straits. Because over here in Asia, high path AI and low path AI is more or less endemic constantly, and those migratory waterfowl will pick up the virus from infected poultry and be carrying that wherever they fly. If we look at the four flyways over the United States, this is just to demonstrate right here that the flyways, there's intermixing and interchange of birds in all four of these flyways, so there's a possibility of the contaminated birds to transmit that virus to all areas of the United States through these flyways. If we look, this is a very, very important diagram demonstrating right here, as I said before, in Asia, which includes China, Taiwan, South Korea, all these areas in through here, where avian influenza is more or less endemic, the migratory waterfowl pick up the virus and they interact over the Bering Strait with the Western Hemisphere waterfowl, and that's how it's transmitted from the Eastern Hemisphere to the Western Hemisphere. If we look at the diagnosis, there are two primary ways in which avian influenza is diagnosed. First is through direct detection, through the antigen detection, through the antigen capture immunoassay, or through real-time PCR, or the hallmark, the standard means of identifying a high path AI virus. That's the gold standard. Standard is that virus isolation and identification, which is done primarily at the National Veterinary Services Laboratory in Ames, Iowa, in the United States. The other way is indirectly through the antibody detection. Antibodies are a protein molecule that are formed upon exposure to a pathogen, and birds that have been exposed to avian influenza will develop antibodies. So we have three primary immunoassays that detect antibodies. Most of the commercial companies in the United States use the agar gel immunodiffusion test, which they collect blood samples from a flock and run those samples for the detection of antibodies, or they can use the ELISA test or the hemagglutination inhibition test. These are the three primary serological immunoassays to detect avian influenza. If we look at a general overview of the history of highly pathogenic avian influenza since it was discovered or identified in the 1890s, we've had several. Obviously, we can see several countries have been affected by highly pathogenic avian influenza. The blue areas are those in which the United States has been infected. In 1927, 25 and 97 was the first occurrence or known occurrence of what they called foul plague back then, or an avian influenza, and this was associated with a live bird market back in the New Jersey area. The next outbreak was in 1983 and 84 in the central part of the United States, the serotype H5N2. That's in the United States. Then we had another incursion in southeastern Texas, and we'll go into these outbreaks a little bit in more detail later on. And the 2015 outbreak, H5N2, and there was one flock infected with H5N8 in the United States during that outbreak. Then in 2016 we had one outbreak involving H7N8. In 2017 an outbreak primarily in Tennessee in H7N9, and then we had one outbreak just last spring in South Carolina involving serotype H7N3. So if we look at that outbreak in Lancaster County, Pennsylvania, that's central Pennsylvania. This is the first outbreak that I was personally involved in. This is an example of the H5N2 strain of that virus was carried by live bird markets in that area, and that particular virus spread to backyard flocks in that particular area, and it happened to mutate from low path AI into high path AI. All this is in Lancaster County, Pennsylvania. In this particular part of the state, there were a lot of backyard flocks, particularly of the Amish ethnic group, and those folks were very interested in controlling diseases, but they had to be, since we had to use good diplomacy to convince them of the problem. So that was our first exposure to dealing with both from the commercial side. The laying egg operations were infected. Some broiler flocks were infected. No turkeys at this time. So at that time in 1983, USDA spent $63 million. There were 443 flocks disclosed involving 17 million birds. Then we look at the high path AI disclosed in 1984 or 2004 in Gonzales, Texas. In this particular case, this is an interesting case. This was a private broiler production area. In other words, this particular gentleman was not affiliated with any particular company, but epidemiologically, this gentleman there are live bird markets in the Houston area, and he was going to the live bird markets in the Houston area, purchasing birds and bringing them back and introducing them into the flock. So epidemiologically, that through circumstantial evidence, that was where that infection originated. In this case, there was no spread at all. This is the only flock that was infected. It was rapidly depopulated, and no further spread was identified. No human infection disclosed. If we look at advance forth to 2014 and 15 to the H5N2 high path AI outbreak in the United States, this was a very severe outbreak in terms of the number of flocks that were affected and how widespread the infection was. So we had a total of 232 flocks involved, 211 of which were commercial, only 21 backyard flocks. A key element here we see Minnesota had 110 flocks affected. 105 of those were commercial turkey flocks. 4 were table egg producing flocks, and only 1 backyard flock was involved in this area. The reason being, Minnesota is the #1 meat turkey producer in the United States, so you can see how devastating that incursion was to their economy. Then we look just south of Minnesota here, we see Iowa with 77 flocks affected, 35 of which were commercial table egg flocks, and another 35 were commercial turkey flocks, and there were 7 flocks that were backyard flocks. And we see down here in Minnesota, we had 2 commercial turkey flocks and 1 backyard flock affected. In Arkansas, where I'm located, we only had 1 commercial turkey flock affected. The infection started over here along the Pacific Flyway in January, where the first incursion was at a raptor rehabilitation center right there, just across the border from British Columbia, Canada, and they were feeding hunter-killed ducks to their the falcons that were being rehabilitated in that particular facility, and they were dropping over very rapidly because it's so hypathia is so virulent in raptors, particularly falcons, hawks, whatnot. Then it spread to 4 backyard flocks here, and 2 backyard flocks in Oregon, and 2 commercial flocks here in California. All this was in January, and we thought it was over, and we had 1 backyard flock in Idaho. Then in March, we thought it was over, but we had the first affected commercial flock disclosed in Minnesota in the first week of March, and almost simultaneously 2 flocks in Missouri and the 1 flock in Arkansas were also disclosed, all of them commercial turkey flocks. So if we look at the key information involved with that 2015 outbreak, 21 states were affected. That included states in which migratory waterfowl carrying that virus were detected. Like I said before, there were 232 premises positive, 211 of which were commercial and 21 backyard flocks. There were almost 50 million birds, commercial poultry involved, 7.5 million turkeys, and 42.1 million chickens. These were all commercial laying hens. Some of those table egg flocks, caged layers, consisted of up to 4 million laying hens in one facility, so that's why that number of chickens mounted up to be so high. So overall, the USDA spent almost $1 billion on this response. So taking all these factors into consideration, this particular outbreak is the most severe foreign animal disease incursion in the United States, in the history of the United States. That includes all species. So that's a very significant factor. So the key here is biosecurity, and we'll go into that a little bit later. The number of counties that were affected, whether it be migratory waterfowl or backyard flocks or whatever, the counties were spread, in the west and in the central United States. The eastern United States were not affected. This is an epidemiological curve illustrating the infection as it was disclosed on a timetable from December through June here. So we see the blue, dark blue columns here are migratory waterfowl that tested positive for that particular strain, H5N2, and then the light blue here was the raptor rehab center that showed up with infection, and then the very light blue with the backyard flocks. So you see how it progressed a long time, and then we through January and February, then we get into March here, the first backyard commercial flock was a turkey flock in Minnesota that was disclosed, along with the 2 commercial turkey flocks in Missouri and 1 in Arkansas, all within the same week. And then once it got into the commercial industry in both Minnesota and Iowa, it spread like wildfire. And that was because of human involvement in terms of transmitting that infection from one place to another through contaminated equipment or service personnel, feed trucks, whatnot. You can see how rapid it spread from one commercial flock to another in Minnesota, Iowa, Wisconsin, South Dakota here. So this is the take-home message here is biosecurity was the responsible for the rapid spread of infection here once it got into the commercial flocks. So once again, widespread. All this infection was due to widespread deficiency in stringent biosecurity, which resulted in rapid transmission. Bio, if we break that word down, biosecurity, bio means life, security means protection. So the reason for biosecurity is to employ, adopt all the measures to prevent contamination or exposure of individual flocks to the virus. Biosecurity, it's a way of thinking, it's a way of life. It isn't, so it's very, very important, whether it's a commercial flock or a backyard flock, that the poultry producer think in terms of biosecurity. So if we look at, we expected the outbreak to continue in 2016 too, because we had such a severe outbreak in 2015. But in 2016 we had one outbreak, H7N8, North American lineage. North American lineage, this particular serotype was isolated by a migratory waterfowl in the area, and through either direct or indirect exposure, turkey flocks in one control area were infected. There were 9 farms infected by contaminated personnel or vehicles, but in only one of these flocks, it mutated from low path AI into high path AI. So we move on to 2017. A different serotype. Once again, North American origin, H7N9, carried by migratory waterfowl in the area. And in one control area, in this particular outbreak, there were 4 states involved, but in one control area in Tennessee, 2 commercial broiler flocks 1.2 miles apart from each other mutated from low path AI into high path AI, H7N9. And there were only 2 cases of high path AI disclosed during this outbreak here. And we had one game bird flock, low path AI, H5N2 in Idaho that year that was disclosed, and one turkey flock, low path AI, H5N2 in Wisconsin disclosed. But the key here are these only 2 affected flocks in Tennessee in the same control area. Then we move on. We didn't have any problems until just last spring in 2020 in North Carolina. We had a low path AI commercial turkey flock H7N3 show up with infection, and it spread from one place to another through contaminated equipment or movement of infected or contaminated equipment or service personnel from one place to another. It got into one flock just across the border into South Carolina, and in one house, this particular facility had 4 commercial turkey houses. In one of those houses, it mutated into low path AI into high path AI, and just one flock was infected. So you can see how important the mutation of particularly serotypes H5 and H7 can occur from low path AI into high path AI to cause a problem. So if we look at various strains of high path AI are endemic internationally in several countries here. H5N1 in 8 countries in 2016, H5N8 in 18 countries in Europe and Asia. Then in 2017, we had various strains involved here. This particular flock here in Mexico central Mexico, in Jalisco, Mexico. This particular H7N3 high path AI got into the commercial layer industry in Mexico, and that is still ongoing now. They've tried everything in the world in terms of including vaccination to eradicate that disease incursion. Then in 2018, once again, a wide variety of high path AI serotypes were involved in a number of countries. Same way in 2019. Then we look at in 2020, just last year, in Europe had a very severe H5N2 high path AI outbreak in several countries here. Germany was the most severely infected country in the European Union. Hungary was the second most severely infected country. So we had several countries in the European Union affected by high path AI. And if we look at a map here in 2020 we have that one flock in South Carolina, over here in the Western Hemisphere, South Carolina, but we have a multitude, hundreds of flocks here in Europe, primarily Europe, but also in Asia, a few in Africa, but Asia and Europe were badly infected. So we had a total of over 12 million birds affected, and there were 625 outbreaks in Europe alone, consisting of a wide variety of serological types. So this is just indicates that high path AI is endemic in certain parts of the world. So the key message is the perpetual risk of high path AI due to wildlife, migratory waterfowl is perpetually present. The only preventive tool is the implementation of rigid biosecurity measures to prevent an incursion. And so in the United States particularly, since 2015, we've only had spotty incursions of high path AI in various areas in which we didn't have spread, but we must continuously combat incessant complacency about not adopting rigid biosecurity as a protective measure. So in terms of repelling the risk of high path AI, whether it be in the United States or worldwide, it's not a matter of if, but when potential exposure to high path AI H5, H7 will occur. That is a fact of life. So we must be prepared to ward off an incursion and further spread by ensuring that stringent biosecurity measures are in place. That is a key for all types of poultry production, whether it be backyard or commercial. So biosecurity is the key principle here. As a result of the 2015 outbreak, the National Poultry Improvement Plan, NPIP, developed 4 biosecurity principles that they promote in the commercial industry and in the backyard industry. But the NPIP was derived in 1935 to improve the health and vitality of the poultry industry, and has progressed from there. NPIP is purely a voluntary organization consisting of private producers, the state departments of agriculture, and the USDA. And they developed 7 or 14 biosecurity principles that should be adopted by commercial poultry flocks. And this is just an overview of all the biosecurity principles. Every commercial flock should have a biosecurity coordinator, because biosecurity is so important, and there should be annual biosecurity training. There should be a line of separation between the poultry facility and the outside area, and a peripheral buffer area within the poultry facility. Site-specific personnel with personal protective equipment must be worn every day. Wild bird and rodent and insect control must be implemented. Cleaning and disinfection for equipment and vehicles must be in place. Proper mortality disposal must be accomplished. Rather than just throwing dead chickens or turkeys out in the field, they must be properly disposed of. And the replacement poultry for either the commercial flock or backyard flock must come from areas that are known to be disease-free. Feed and litter replacement must be controlled. Water supplies must be disinfected, preferably from a well or a municipal source, because water can serve as a vehicle for contamination from migratory waterfowl. Disinfected feed and litter must be continually applied. Proper mortality is disposed. Reporting of elevated mortality and morbidity. And these facilities are audited every other year by official state agencies, the State Department of Agriculture. So these are just the 14 biosecurity principles that have been developed and promoted by the National NPIP in the United States. So the conclusions, it's not a matter of if, but when high path AI will reoccur in the United States, because high path AI is rampant in Europe and Asia. The wildlife reservoir, migratory waterfowl, is omnipresent. Biosecurity is the primary preventive measure, and the battle against complacency is perpetual in the United States as well as worldwide. So that completes my presentation on how devastating highly pathogenic avian influenza can be to the poultry industry. Next, I will discuss virulent Newcastle disease.